They have 8 arms, each covered in suckers, and those suckers can both taste and feel their environment. So they're always exploring, they're always moving around, and they almost never stay still enough to show you that third right arm, let alone show you if it has a hectocotylus, because they're trying to get away from you and explore that environment. And that leads to the first problem of sexing an octopus phenotypically. It is really hard to find that third right arm, even in captivity. So imagine if you're diving in the ocean, the octopus is moving around in the water or on the ground, you're probably not going to be able to quickly find that third right arm with that hectocotylus present or not. And that is a huge problem. Also, keeping an octopus still enough to look for that third right arm causes a lot of stress. They're constantly tasting and feeling. They don't want to be held still for you to uncoil that arm to see if they have a hectocotylus on it or not. But there is a secondary characteristic we can look at to identify male or females, and that's enlarged suckers. The octopus on the right is a male, and there's an arrow pointing to that enlarged sucker, while the octopus on the left is a female. All of her suckers are pretty uniform. This is pretty easy to spot, but it's not completely accurate. This is pretty similar to relying on human breast size or facial hair to determine the human sex. Some male humans might have more breast tissue than some female humans, and some female humans might have more facial hair than some male humans, and that does not determine their overall sex. That's the same here. Some male octopuses might have that enlarged sucker, or they might not, and some female octopuses might have uniform suckers, or they might not. So it's not really all that accurate to rely on this secondary sexing method. And also, in the wild, you're probably never going to see an octopus in this position. They're not really going to show you their mouth parts with their suckers all displayed like this. You can really only see this in captivity with a glass container. The last problem with octopus sexing is it is actually impossible to sex a baby octopus because they do not have a hectocotylus or enlarged suckers at all. They don't develop those traits until they're fully mature. So in that vein, you also cannot sex an octopus in an egg because they also don't have those characteristics. So there's a whole lot of problems with our current methods of trying to figure out if an octopus is male or female. It's really hard to find that hectocotylus, and it's actually impossible to sex them until they're mature. So why is this important? With our current political climate and research defunding climate, sexing octopuses might not seem like the most important thing, but it actually is. The genetics field as a whole is based off of a few model organisms, like those fruit flies, those mice, those rats. So a lot of genetics is based off of a few animals. So by looking into non-model organisms, like octopuses and cephalopods, you're answering a whole lot of questions for the entire field of genetics, because you're looking at an animal that isn't often studied. And like I said earlier, we didn't know any genetic sex for any cephalopod. So specifically looking into octopus sex would not only open the doors for genetics as a whole, but cephalopod genetics also as a whole. And this is really important because there is some evidence that because of climate change and ocean acidification, octopus populations might eventually start decreasing due to those changes. And usually, when an animal's population is decreasing, we do things like captive mating efforts to try to increase their populations. And we would do that for octopuses because they're found in almost all depths, in almost all marine habitats, and they're really important prey items for birds, for fish, and also for humans as well. So if their populations start decreasing, then a whole lot of other negative things will start happening in the ocean. So being able to genetically sex them by taking a little sample, figuring out if they're male or female, will help with mating efforts as we can pair octopuses together, but it'll also help with things like sex ratios. In climate change scenarios, a lot of the times increasing temperatures or increasing acidification messes up the ratios of animals. So instead of 50-50 male and female, there's a chance that there might be more males being born or surviving or more females being born or surviving. And that's important to know because that means they're not going to be making as many octopuses if their ratios are off. So being able to get samples of the larvae and the eggs and being able to genetically test them to see if there's any sex ratios being affected as well is also really important in regards to climate change. So now that we know that it's actually really hard to sex octopuses phenotypically, and it is important to do this, let's get into the nitty-gritty of sex determination. Like I said, we did not know the genetic sex of cephalopods, so I really had nowhere to really start for this project. So I had to start by looking at the most common type of sex determination, which is chromosomal sex determination. chromosomal sex determination was first discovered by Nettie Stevens in 1905. She was looking at mealworm chromosomes, and from looking at their chromosomes, she found that male mealworms had one chromosome that was significantly smaller than all of the others, while female mealworms had all of their chromosomes around the same size. And these chromosomes later became known as the X and the Y chromosomes. In this system, males have that one small chromosome copy that's significantly smaller. It has way less genetic information than the X copy, while the female has two copies of the same chromosome, the X, and it's around the same size as all of the other chromosomes. This system is known as male heterogameity, meaning males have one copy of that unique chromosome while females have two. And it's most commonly found in mammals and some insects, but it's also found in humans like you and me. In contradiction to that XY system, there is also the ZWZZ chromosomal system. This system is really similar. There is still one chromosome that's significantly smaller, but in this system that's denoted as the W chromosome. And the females have that chromosome that's a lot smaller than all of the others. So they're ZW. Males have two chromosomes that are around the same size, and that is ZZ. This system is known as female heterogameity because females have that one different chromosome copy while males have two of the same. And this system is most commonly found in birds, but it's also found in some snakes and also some insects. The last chromosomal system I'm going to be going over is the XO and the ZO chromosomal systems. These are really similar to those last two I just went over, but instead of having a really tiny chromosome, animals with this system are just completely missing that tiny chromosome. So in XXXO animals, the female is still XX and the male is missing that Y, which is denoted as XO. In the ZO system, males are still ZZ, but females are missing that small W and is denoted as ZO. And this figure is just showing how this system is inherited. There are some genetic disorders in humans, leading to a human to be missing one of their sex chromosome copies, which causes infertility. But in animals where this system's pretty common, that lack of a chromosome just gets carried down as if it was a Y chromosome, leading to this system continuing to occur in nature. So now that I went over the most common types of sex determination, I had to learn about all of the other types, just in case octopuses weren't that common. And so I'm going to be talking about non-chromosomal sex determination. The most common type of, the first non-chromosomal sex determination I'm going to be going over is sequential hermaphrodism, which I think is the most common type. And this is when an animal's sex can switch at some point in their life. There's three different types of sequential hermaphrodism. There's protandry, which is switching from male to female. There's protogeny, which is switching from female to male. And there's bidirectional, which is when an animal can switch in either direction, and sometimes that can happen multiple times in that animal's life. I think the most common example of sequential hermaphrodism is seen in clownfish. Clownfish use the protandry system, and that means that all clownfish are born male. And clownfish live in little groups with one female and a whole bunch of males. In those groups, if that one female dies, or if she swims off and joins another group, the largest male will have his testes degenerate and latent ovary cells develop into ovaries. Once those ovaries are developed, that clownfish is now a female who can now mate with the rest of the males in that group to create more clownfish. So now that you know that, if you ever watch Finding Nemo again, now you know that Nemo's mom died, so his dad is probably very slowly turning into his mom throughout the duration of the movie. Another example I'm going to talk about is temperature sex determination. And this is a sex determination method that relies on the temperature in which the eggs are incubated. I'm going to talk about two patterns, the MF pattern and the FM pattern. The MF pattern is most commonly found in marine turtles, with the FM pattern being most commonly found in crocodiles. In the MF pattern, eggs incubated in low temperatures have a higher chance of developing as male, while eggs in high temperatures have a higher chance of being developed as female. The opposite is true for the FM pattern. Eggs incubated in low temperatures have a high chance of being developed as female, while eggs in high temperatures have a high chance of being developed as male. Now, this system was believed to only be found in those turtles, lizards, crocodiles, and alligators, but it was recently found that this system might be found in some birds as well. This is the Australian bush turkey, and it buries its eggs in mounds in the ground. And in this species, it was found that eggs incubated in low temperatures will develop as male, while eggs incubated in high temperatures will develop as female. So really similar to that MF pattern in marine turtles. Now this is interesting because it shows that a field like this is constantly discovering new information. So nothing is concrete in systems like this because we will always find something new, like this Australian bush turkey. There is also location-dependent sex determination, and I think this is pretty self-explanatory with its name. So where the animal is located causes its sex to form. This is most commonly found in marine animals, and the first example I'm going to talk about is the marine worm, Bonellia viridis. So in this worm species, larvae are born undifferentiated, so they are not born as male or as female. And as they're swimming around in the ocean or crawling around on the ground, if that undifferentiated larvae comes into contact with a female, it will develop male reproductive parts, because females produce a chemical called bonolin, which triggers those reproductive parts to grow for males. If that larvae never comes into contact with a female, it will either remain undifferentiated until it does, or it will just turn into a female to create more of that bonolin, to create more males for the worms to mate with. There are also slipper snails, which is a pretty common example of this system, and slipper snails form mounds on the ocean floor and they kind of just live in piles like that. Like clownfish, they're all born male, but unlike clownfish, they have a life phase where their entire reproductive system degenerates, so they become undifferentiated. Once they're undifferentiated, if they're attached to a female in that pile, they will revert back to being a male. If they are removed from the pile, they will become a female, and if there are too many males in the pile, they will become female. And in this species, becoming a female is completely permanent, but if they are male, they can become undifferentiated again. So they can either become a male or a female again. So where they are in this pile completely determines whether they will permanently become a female or whether they'll temporarily turn into a male. The last method I'm going to be going over is SNP-dependent sex determination, specifically with tiger pufferfish. Now, a SNP is a single nucleotide polymorphism, which is just a variation in a single nucleotide in a specific part of a genome. In tiger pufferfish, there's a specific SNP in the anti-Mullerian hormone receptor type 2 gene that affects a really specific amino acid. And this change affects the amount of times that amino acid is seen in the genome. So if that amino acid is seen twice, then that creates a female pufferfish because they're homozygous. If that amino acid is seen once, then it becomes a male because that amino acid is only caused once. So the sex of a tiger pufferfish is completely dependent on that single nucleotide change, which affects how often they have that amino acid in their genome being produced, which causes their phenotypic sex to be developed. So now that we know all about some of the most common sex determination systems, the question still remains, what do octopuses have? I've said already that we don't know anything about cephalopod sex, so I really had to start from scratch. From looking at all the non-chromosomal sex determination systems, it was pretty clear that octopuses probably don't have those. We've never seen an octopus drop its hectocotylus to become a female. We've never seen a female develop a hectocotylus in the lab. So far, we haven't seen any temperature or location dependence triggering their sex. So they probably don't have those non-chromosomal sex methods. With that knowledge, I then looked into chromosomal sex methods. There was nothing involving cephalopods, so I had to look at the next best thing, which is the phylum Mollusca, and octopuses are in phylum Mollusca. There were a few big researchers looking at non-hermaphroditic mussels and what their genetic sex is. And for the non-hermaphroditic mussels, it was found that they most commonly used the XXXY chromosomal system. So with the knowledge that I knew that octopuses probably are not hermaphrodites, they probably do not rely on temperature or location, and because I knew that mussels use an XXXY system, I ran off that knowledge. So my hypothesis was that octopuses probably have an XXXY system. So how do you even begin looking into the genetic sex of octopuses? The first step is to get octopuses. So my divers got 28 octopus rubescents from Driftwood Park in Washington. 10 of those octopuses were female and 18 were male. These octopuses are found in glass bottles on the ocean floor, and the divers find those glass bottles, they put them in Ziploc bags we brought up to the surface, and we dump them into red nalgenes. And this video is just showing an octopus entering that red nalgene. Once an octopus is in that red nalgene, we can then bring them back to Rosario Beach Marine Lab, where they are stored in tanks with flowing seawater and that red nalgene for cover. When they were there, I fed them either a purple shore crab, a purple whelk, a mussel, or a manila clam daily. And once I was done with my experiment, if they were happy and healthy, I returned them back to Driftwood Park. To get DNA from any animal, you need a sample of them. And in my case, I decided to take a little bit of their arm tips. To humanely do that, I had to sedate the octopus. And to sedate an octopus, you slowly add ethanol into a container that they're in. And this is a video showing that. So this is a pretty large male who took around 35 minutes to become fully sedated while adding the ethanol. As you can see, as I add the ethanol, he stops moving around as much as he was, his coloration stops changing as quickly as it was, and towards the end, he kind of just flops over. At that point, I would have touched him, and if he was non-reactive to touch, I would have continued. If he did react to my touch, that means he was not fully sedated, and so I added more ethanol until he was. Once the octopus was fully sedated, I cut 2 centimeters from 2 of their arms. So I had 2 samples per octopus, and those were just stored in microcentrifuge tubes in the deep freezer until I was ready for DNA extraction. I got a lot of help with my DNA extraction, which I really appreciate, because it was a lot of work. The first step we had to do was defrost each of those arm tips and peel the outer layer of skin off. This is because the skin has a lot of bacteria on it. And I don't care about bacterial DNA or RNA, I only care about octopus DNA. So I did not want any microbial contamination in my DNA. So once that skin was peeled off, we then finely chopped that muscle tissue and refroze it until I was ready to actually extract the DNA. I used a Monarch Genomic DNA purification kit and followed the instructions to extract the DNA from that peeled, finely chopped muscle tissue. And once I had my extracted DNA, I sent it off to a company called Admira Health. And Admira Health did double digest restriction site associated DNA sequencing, otherwise known as dd-RADseq. And dd-RADseq is a type of high throughput sequencing that sequences a large number of sample locations throughout an entire genome. These sequences are pretty shared between individuals, meaning I'm getting a lot of the same areas of the genome for all of my individuals. This method is really cheap and it's really fast, which is perfect for my project. They did dd-RADseq via Illumina sequencing, and they used 2 restriction site enzymes, EcoR1 and MSP1. I picked these restriction site enzymes because there was a published paper where a scientist extracted the DNA of a closely related octopus species using these restriction site enzymes. And I wanted my project to work, so I used those restriction site enzymes since they've already been proven to work. The first step of dd-RADseq is for that first restriction site enzyme to start to cut the DNA. Now, restriction site enzymes find specific DNA sequences on the entire genome. These sequences are pretty short, they're around 4 to 8 base pairs long, and they're found hundreds of thousands of times throughout the entire genome. So they find them in a whole lot of places. My first restriction site enzyme found every spot that was GAA-TTC, and it cut between the G and the A, and it did this for all of those sequences throughout the entire genome of each octopus. The second step is to add adapters, which is important for Illumina sequencing. And the third step is for that second restriction enzyme to start cutting the DNA. MSP1 looks for CCGG, and it cuts between that first C and that second C. Again, these sequences are found hundreds of thousands of times throughout that genome. Once that happens, more adapters are added, which is important for Illumina sequencing. And then I had my inserts or my fragments, little pieces of DNA cut away from that whole genome, and I will refer to these as rad tags. A rad tag is just that insert or fragment that has genomic information that I'm looking at, that I'm looking for. Inserts are typically around 250 base pairs, that's just the industry standard. So my inserts, I kept them at 250 base pairs in length. In total, I had 16.2 million total raw paired end reads, ranging from 194,000 to 1.5 million per octopus. Now I said those reads were paired end reads. That basically means that there's a forward strand and a reverse strand that reads that insert. That forward strand reads the DNA from left to right, and the reverse strand reads the DNA from right to left. At some point when reading, there is a period of overlap because it's reading that same insert. I wanted to merge at those 50 base pairs of overlap, and this just overall increases the accuracy in the read because you can find any errors in those reads, so if they don't match up, you know there's an error there and you don't want that sample. So this increases the accuracy, and it also creates longer reads. So instead of just having 150 base pairs that aren't all accurate, I now had more accurate 250 base pair long reads that I can use. So after merging, I had 13.7 million total reads remaining. So I lost around 3 million, which is normal for this process because of the errors in that read sequencing. There was merging errors, they were too big, they were too small. So just something happened there, and that's pretty normal and expected for this method. So now that I had all of my fragments, all of my rad tags, I actually had to look inside of them to find my answers. There were 5 things I did here. I used a program called Trimmomatic, then I tried to use a program called RadSex, which did not work, which made me use my last 3 methods to try to find answers here. So that first thing I did was I used that program called Trimmomatic. Trimmomatic cleans the reads, so it gets rid of any of the base pairs at the end of each of those sequences that are really low quality. And this happens pretty often because of how Illumina sequencing works, and that's normal. So it just cuts away those reads that are really low quality, or it completely gets rid of an entire read that is just really, really bad. I don't want any low quality reads in my data because that would make my data less accurate. After quality control trimming, I had 13.5 million total reads remaining, so I lost around 200,000 just because they were really low quality. I next tried to use a program called RadSex, which was developed in 2021. This was to try to find any of those rad tags that were associated with sex. So whether there were specific rad tags in males or specific rad tags in females. Unfortunately, this did not work for me. I did not get any results. So instead of my project ending at 2 steps and giving me a nice clean answer, I had to manually search for those rad tags myself. And by manually searching for those rad tags, I was looking for the presence or absence of unique rad tags in males or in females. So whether males had unique rad tags that females didn't, or if females had unique rad tags males didn't, and anywhere in between. To do this, I had to create a library to create a reference file. So I used my octopus with my male octopus with the most sequencing, which was octopus 7, and my female octopus with the most sequencing, which was octopus 25. I combined those sequences into a single file, and I clustered any similar reads together. Now, a lot of the reads are similar because these animals are from the same species, so of course their DNA is going to look pretty similar all the way around. So if their reads were 97% similar, I just clustered them together, so they're one specific read. So from doing this, I had 254,000 representative rad tags to work with, which makes up around 54 million base pairs, or 1.4% of the entire ovirbescence genome. You may be looking at that number and being like, 1.4%, that's really, really tiny. How are you going to get an answer from that? And let me give you an example here. So let's say that all of the men in the United States disappeared. They went to one of the 50 states, and I'm the only one that has to figure out where they all went. And the most expensive and time-consuming way of figuring out where all of the men in the United States went is to travel to each of the 50 states and ask each person if they are a man or a woman. This would take a whole lot of time, a whole lot of money. So instead of doing that, I could send out a random survey to 250,000 people spread randomly throughout the United States and ask them in that survey if they are a man or a woman. And this is looking at less than 1% of the US population. But by doing that, there would be one state that pops up as there's a bunch of men there. So I found the answer of where all of the men are in the United States. So by looking at 1.4% of the ovirbescence genome, I was basically doing the same thing. I was randomly surveying that genome to try to find if there are any rad tags specifically present in males or specifically present in females. Once I had those clustered reads, I then had to align each of those individual octopus genomes to that reference file. And once I did that, I had 13.3 million total reads mapped to those specific rad tags. I could then count the amount of rad tags that were present in males and present in females and see if there was anything significant there. I had some really interesting results, and I visualized them in this heat map here. So the x-axis is my amount of males. I had 18 total males. And my y-axis is my amount of females, so I had 10 total females. The more red the color is on this heat map, the more unique rad tags were present. The more white or blue the color is on the heat map, the less amount of rad tags were present. So let's walk through how to read this with just a random non-significant square, like this one right here. This square is kind of a lighter orange, which looking at that key in the corner represents 2,000 to 5,000 unique rad tags. So looking at just this square, there are 2,000 to 5,000 unique rad tags found in 8 of my males and 3 of my females. So there are that many rad tags shared between 8 of my males and 3 of my females. And that's just how you look at this chart. So trying to find significant answers was really interesting. The first interesting thing I noticed was there are actually 0 rad tags that all of my males had and none of the females had. So there is not one significant rad tag that only males have. And the same can be said for females. There is not one rad tag that all of my females have that none of the males have. So there is no sex-associated rad tag in octopuses as far as I could tell with these results. And looking further at this figure, you can see that I actually had nothing even close to a sex-specific rad tag. There's a whole bunch of white and a whole lot of blue, meaning that there's nothing really close to that. The closest we can get to a sex-associated rad tag is 1 to 5 unique rad tags present in only 12 out of 18 of my males that none of the females had. 1 to 5 is a really low number. That's basically nothing. The same can be said for the females. The best we can get is 1 to 5 unique rad tags found in 8 of my females and none of the males. So again, there is no specific sex-associated rad tag for octopuses, as none of my males had a specific and none of my females had a specific one. But there was one hopeful little data point, and it is this really, really dark square right here. This dark square represents over 20,000 unique rad tags, meaning my females, there was one female that had over 20,000 unique rad tags that none of the male octopuses have. And that is a really big number. None of these other squares are even close to over 20,000 unique rad tags. And this kind of provided a little bit of hopeful evidence that females might have something funky going on that the males don't have. They have over 20,000 unique rad tags that none of the males have. And that's really interesting. So the next thing I looked for was trying to see if there were any genetic variations that were sex-associated as well. And I looked for 2 things: the SNPs, which is really similar to those tiger puffer fish where they had one SNP that determined their sex, and also indels. So I was looking for inserts and deletions. An insert is just when a nucleotide is added to the DNA sequence, and a deletion is just when a nucleotide is deleted from that DNA sequence. So I used a program called Plink2 to see if there were any sex-associated SNPs or indels, and I did not find anything here. So octopuses are probably not like tiger pufferfish. They do not have a specific SNP or a specific indel that triggers their sex to develop in a certain way. And the last thing I looked for to try to find any really significant results was the rad tag count analysis. And this is counting the amount of unique rad tags present in each octopus. So instead of presence and absence, I was now counting the amount of unique rad tags. To do this, I used a program called My first significant rad tag closely matched to a species of bacteria. And again, I don't care about bacteria, I care about octopuses, so I completely just disregarded that rad tag because it was from contamination. But my second significant rad tag closely matched with O. bimaculoides, which is an octopus species that's closely related to Ovirubescens. And it matched with that with around 85% identity, which is huge. The reason it didn't match to Ovirubescens is because Ovirubescens isn't in BLAST. We don't have a genome for them in BLAST, so it matched to the next closest thing it could get, which is O. bimaculoides, which meant that that rad tag was my octopus genomic information. So this is a visual of that rad tag count, and it is in a bar graph here. So the X is the sex, so the first bar is female, the second bar is male. The Y-axis is the amount of times a specific read matched with a rad tag. And all of the dots represent individual octopuses. So my female bar has 10 dots because I had 10 females. My male bar has 18 dots because I have 18 males. And that really dark black line in each of those bars represents the average amount of reads that map to that rad tag per octopus. So looking at this figure, specifically the females, I had 7 out of 10 females have reads matched to this unique rad tag. Out of all of those females, I had 1 female have 37 reads mapped to that rad tag. In total, in my females, I had 106 reads mapped to that specific unique rad tag, giving me an average of around 10 reads per female octopus for that rad tag. My males, in comparison,
I only had one male have one read mapped to that rad tag, which means the average amount of times that rad tag is found in males is 1/18, which is basically 0. So this rad tag is more commonly found in my females, 7 out of 10 of them had it, and there was 106 reads in my females matching to that rad tag. Once again, kind of suggesting that females might have something funky going on, because the males didn't really have this rad tag present at all. So what do these two results mean? Like I've been saying, these results kind of point to females having something funny happening, which would be most closely aligned with female heterogameity, which is that ZWCZ chromosomal system, where females have that different chromosome. And this can be seen in that one figure with the females having all of those reads for that one specific rad tag, and the males not having it, which would suggest that females have a unique chromosome that the males don't have. And again, it can be seen here with females having 20,000 unique rad tags that males don't have, suggesting they have that W chromosome. But overall, my results were really inconclusive. I had 250,000 rad tags mapped to 13 million reads, and octopuses have 30 chromosomes, meaning if there was a specific chromosome determining sex, I would have found thousands of rad tags present in one sex and absent in the other. Instead, I only really had one rad tag that was kind of unique, and then 20,000 out of one of my 10 females, so these results don't really show anything specific here. But one of the super cool things about doing research is it means there's probably somebody with a little more money trying to answer the same exact question as you. So when I started this project, I unintentionally entered the space race equivalent of genetic sex determination of octopuses with University of Oregon. And unfortunately for me, University of Oregon just had a little more money and a little more time, and they got to the moon first. They published a paper regarding genetic sex determination in February of this year, right before my project wrapped up. Which sucks for me, but it's super cool for science as a whole. Gabrielle Coffing from University of Oregon used cool and expensive methods like PacBio's long high-fidelity sequencing and chromosomal conformation capture to basically find out octopuses probably have a ZOZZ chromosomal system. This is because she found that Ovimaculoides have 2 copies of chromosome 17, the males do, and the females have 1 copy of chromosome 17. She also found 1 large sex chromosome in other octopuses, in cuttlefish, in squid, and in nautilus, so all of those other cephalopods. So these results are really probably the correct result. She had really good methods, very expensive methods, really good data, and she found this answer in almost all cephalopods. It would be very strange if ovirubescens was different from all of these cephalopods, and that's probably not the case. Because of how closely related ovirubescens is to Ovimaculoides, her main octopus, ovirubescens are probably also ZOZZ, and it would be very, very strange if that was not the case. So why did I find a W chromosome instead of a lack of a chromosome? Those seem like 2 very different things. And that is because of the methods I used. So ddRADseq is really, really good at finding sequence differences between populations, but not chromosomal copy number differences, which is the problem here. So females are missing that chromosome, and ddRADseq could not find that. There was nothing to compare the data to because they are missing that chromosome copy number. And I used ddRADseq because a ZO or XO system is incredibly rare. I didn't even think that this would be possible in octopuses because it's just not found too often, and mollusks also have that XXXY system. So this information is from the Tree of Sex database, which is a public database that scientists update almost every day. They update it with their study organism and the genetic sex associated with that organism. So the last time I looked at the Tree of Sex, there was 11,915 published invertebrates with associated sex. Out of those invertebrates, 0.31% had that ZW system, 1% had that ZO system, 17% had that XO system, and 42.28% had that XY system. So the XY system is just really, really common. And because of the evidence that mollusks that are not hermaphrodites have that XY system, I chose ddRADseq to try to find sequence number differences instead of chromosomal copy number differences. Hypothetically, my rad tag count analysis could have shown a ZO system, because this is just counting the amount of rad tags. Unfortunately, because I did not think that this system would be a possibility, I kind of didn't spend my budget correct for the system to work. So I had 28 octopuses, and I got pretty good sequencing results for them, and that's how I budgeted. If I thought it was possible to be missing a chromosome, I would have had way less octopuses and got way better sequencing results for each individual octopus. And this would have allowed me to look at more of those rad tags, count more of those rad tags, and look through the noise to find that missing chromosome. But unfortunately, because I did not think that was possible, I did not spend my budget in the way that would give me this answer. Now, Coffin, towards the end of her paper, did mention it is possible a very, very, very tiny small W chromosome does exist, and she just could not find it because it's just too tiny. So because I found a unique rad tag, I wanted to see if it was possible that this rad tag could have been that very, very small W chromosome. So I took that rad tag and I matched it to that O. bimaculoides genome to see what chromosome it was found on. And it was found on chromosome 2. If you know anything about chromosomes, they are numbered by how large they are. So this rad tag was found on the second largest chromosome in the ovirescence genome, meaning it is most certainly not a very, very, very tiny chromosome that we could not find. So this rad tag maps to a very large chromosome. So even if it is true octopuses have a small .W chromosome, I did not find it here with this one unique rad tag. So overall, I was half right. Males are ZZ, but females are not ZW. It is quite likely females are ZO, and I just didn't use the right methods to find that answer. So even though my data was not really good at answering genetic sex determination in octopuses, it is really useful for other projects. I had 28 octopuses that I got dd-RADseq from. Coffin only had a few octopuses per study species that she was looking at. So I have a whole lot more genetic information, which can be used for a lot of other projects, like looking at introgression or hybridization, comparing individuals between each other, or even seeing if the genetic sex does not match up with their phenotypic sex, because that is always a possibility. So in conclusion, what is the genetic sex of octopuses? It is probably ZOZZ, and I was half right, because I took a bet and my bet did not play out. I would like to thank a lot of people, specifically Alan Birdie and his 2 undergrads who caught my octopuses for me. It's because I am scared of the ocean, so I was not going to do that. I would also like to thank Meg, Riley, Stacy, and Jamie for helping me collect and care for and process my octopuses. Also, thank you to all 28 of those octopuses who let me take 2 of their arm tips. And thank you to my committee for all of their guidance, Dr. Onthank, Dr. Nestler, and Dr. Brothers. This project was dedicated to my master's advisor and also my undergraduate advisor, and every other science teacher I've ever known, because without them, I would never question the unknowns of the world. And also thank you to my family and everybody else who supported me. Thank you. Any questions?
So we're going to have to do something a little bit weird here, is that we will move down to 112 for questions, because GB is actually waiting outside to get in the classroom for general biology to start. So we'll move down to 112, take the questions there, and yeah, move on from there. Thank you.